Hey guys, so I'm going to kind of go through these notes with us just so that you have a second opportunity to hear this again in case you didn't catch it all the first time. So, what we're going to be doing is just normal genetics, normal um, mom, dad, they're going to make offspring. There is a combination of these traits within that offspring. And so, we're going to look at predicting this pattern because again, with all of this, if this is probability, um, so every time that there's offspring produced, the same percentages are going to be there every time. Um, and so we're going to do the ones over a monohybrid cross and a dihybrid cross, but there is the trihybrid cross too, which is 3 traits. But I will let you be able to do that because it's the same concept once you do monohybrid and dihybrid crosses. Um, So this first one that we are looking at is going to be this monohybrid cross-linked at 2 traits. But what I want to kind of show you really quickly is making these gametes and kind of going through this through meiosis. So remember that in meiosis, so say this is mom, we're going to duplicate DNA. So something like this, right? And then we're going to go through meiosis 1, where we split the cells there, and we go into meiosis 2, and because there's no interphase before meiosis 2, we're not going to duplicate the DNA again. Oop. And so it's going to go something like this. And the only combinations we can give the offspring is either this capital R or this lowercase r. And so that is what we're looking at with this trait right here, with this set of alleles right here for this trait. The other thing I want to talk to you about real quick is the difference between traits and alleles. So trait is the characteristic. So in this case, we're looking at the type of seeds, whether it's a round seed or wrinkle seed. The type of seed is the trait. So in this case, capital R would be round seed, for example, would be the trait here. The alleles are my letters, okay? And those are, every trait has 2 alleles. So in this case, a capital R and lowercase r, or a dominant, if I can spell, allele, which is the capital R, and lowercase r would be a recessive allele. And these are what's going to be given to the offspring to make up the traits that make up that offspring, okay? So, let's erase that. So in a garden pea, we have a round seed, capital R is dominant to wrinkle seed. So we know that capital R is round, lowercase r is going to be wrinkled. Heterozygous dominant. So let's look at what heterozygous means, just kind of remember. Heterozygous or hetero means different. Okay? And then homozygous, or the prefix homo, means same. So when we're talking about different, it's going to be a dominant allele and a recessive allele every time for heterozygous, because it's different. Homozygous or homo same is either going to be both dominant or both recessive. Now, those are what we call genotypes. Genotypes are the alleles of that offspring or of the parents. Okay, so we're looking for letters. Looking for phenotypes, we're looking at the trait given off or what you see in a photo. So these right here are alleles, right? And these are going to make up the genotype, while the phenotype would be like this capital R, lowercase r, would be coded for round because it's dominant, it has a dominant trait in there. While if it has 2 capital R's, it's going to be phenotype round because there's 2 dominant traits. The only time we ever see a recessive trait show up in the phenotype is when there are 2 recessive traits or 2 recessive alleles only that's going to code for that recessive trait. So this will count for wrinkled. Okay, so we're going to look at the possible genotypes, resulting offspring, give the offspring phenotypes and their phenotypical ratio, which we'll talk about here in a little bit. So genotypes. Okay, so mom would be capital R, lowercase r, right? So another word for this would be heterozygous dominant, because it's going to show the dominant trait, and they're different, so heterozygous. And then lowercase r for dad, which would be homozygous recessive. Because it's two lowercase, it's recessive. They're the same, so homozygous. So then the trait for mom would be round seed, and the trait for dad would be wrinkled seed, because that's recessive. Okay. So now we're going to get into what we're doing. So what you guys remember in 7th grade was this idea of a Punnett square, which is great. That's awesome. I love Punnett squares, but only for if we're looking at one trait. And so what you guys probably drew was something like this. You had mom, you had dad, you crossed them. Great, that's awesome. Okay. However, it takes a lot of time when I'm dealing with 2 or 3 traits at a time. And when we get to end of course testing, they're not going to ask you for one trait. They're going to ask you for multiple traits. And so we're going to do this mathematical formula that kind of gets us to where we can do this a lot easier with a lot less time, because you only have 75 minutes to take your end of course test. So what we're going to do is we're going to create this gamete formation through meiosis, and we're going to do this through based on the law of segregation and the law of independent assortment. So as we look, when we get to, we're going to talk about, we're going to have step 2 here, then we're going to have step 3. So we're going to talk about these, but the first thing I want to talk about is this law of segregation. So what this means is that there is a random chance that these two alleles are going to separate like this. So that the one on the left is always going to be dominant, the one on the right is always going to be recessive. These alleles are going to separate from each other, and they're not going to be inherited together. It's only going to be one or the other. Okay, and then the law of independent assortment would mean that basically every trait is going to segregate from itself independent of other alleles. So what that means is, let's say that I had mom, and they were both homozygous dominant or heterozygous dominant for these two traits. I don't know what they are. That does not mean that the capitals or the dominant traits are going to be inherited together and the recessives are going to be inherited together. Okay, it could be capital R lowercase f, could be lowercase r capital F. There are multiple ways to do that because they are going to inherit separately, or they're going to independent or assort separately when they go through meiosis, through that gamete formation. So what we're going to talk, that's what we're going to be looking at with this, is this is going to show us the law of segregation. Then we'll look at the law of interdependent assortment here in a minute when we get to the dihybrid crosses. So what we're going to be looking at is what two traits or alleles can we give the offspring. So mom can either give the dominant trait or the recessive trait because she has two options. Now, what is the odds that we're going to get a capital R within the offspring? Well, there are two chances. There's only one capital R, so 1 divided by 2, or 50%. Same thing with the lowercase r. Or what you guys saw here, this shows up in 2 of the 4 boxes. Lowercase r shows up in 2 of the boxes, 1/2, 2 out of 4. While dad is giving lowercase r and lowercase r, the only options we can give lowercase r in every box, so that's 100% or just 1. Okay. Now this is where we get to predicting possibilities. So we're going to do the same thing what we just did, because now we're going to see what one parent is going to fertilize with one of the other parent. And so what we're going to be looking at here is looking at this outcome. Okay, so we have this and we have this, and now we are going to find the outcome. So everything's still the same. And here's what we're going to do. Okay, so what we're going to do is we're going to multiply this probability times this probability, and then this probability times this probability. Okay, so what I mean by that is this: is the genotypes that we can have is capital R and lowercase r from this one to this one, and lowercase r, which is this one to this one. And so what we're going to do is we're going to multiply. 1 times 1/2 is 1/2. 1 times 1/2 is 1/2, or what you guys know is 50%. So that's my genotype. Because remember, genotype is the alleles. Okay, so there's only two different alleles we can have: capital R, lowercase r, and lowercase r, lowercase r. Now the offspring phenotypes is what we see, okay, or we look in that photo. So remember, our capital R was round, and our lowercase r was wrinkled. Now remember, lowercase r, in order for it to show up, has to only be homozygous recessive. If we have this or this, it's going to show up as dominant because there's a capital R in those two alleles within that trait. So this capital R, lowercase r, we're going to add them up together. So there's 1/2 of those because there's only one of those. So 1/2 is going to be round seeds, right? Lowercase r is going to be 1/2 because there's only one of those. Wrinkled seeds, okay? So now here's where we get to dihybrocrosses, or two characteristics or two traits that we're looking at. So we have the same round seed, capital R, and wrinkled seed, lowercase r, and then we're going to look at purple flowers, which is capital F, which is dominant to white flowers, or lowercase f, okay? And so a genetic cross between heterozygous, hetero, different, between a P female and a homozygous recessive male. Okay, homo, same. So we're going to use the probability rules and the tree diagram to show all the possible genotypes resulting offspring to give their phenotype ratio. And so, I'm glad I read that, because now we want to give the ratio of these. So what we mean by ratio is it's usually, when we're looking at monohybrid traits, it's dominant or homozygous dominant to heterozygous dominant to homozygous recessive. And then this is for genotypes. And for phenotypes, you do dominant usually to recessive. Okay, so what I mean by that is homozygous dominant, was there any capital R, capital R's? The answer is no. So we would really just leave it blank, really. Then how many heterozygous dominants were there? It was 1/2. So you could either do 1, and then how many homozygouses were there? There was 1/2, so 1. So there are a number of ways you can do these traits, and here's where I'm going to show you that it doesn't really matter how you do it. So you can write these in percentages, you can write these in fractions, because it's all going to be the same. So there were 0 out of 2 dominants, there was 1 out of 2 heterozygous dominant, and there were 1 out of 2 homozygous recessive. Well, if you cancel out the like terms, or the bottom of the fractions, you're left with 1 and a 1. So a 0 to 1 to 1 ratio. You can also do the same thing with 50%. You have 0% to 50% to 50%. Okay? As long as these two numbers are the same, right? We know that it's a 1-to-1. You can make them 2-to-2, whatever that looks like for you. As long as you can find your answer doing it that way, it doesn't really matter to me. Okay, but I would also suggest if you don't do it heterozygous or homozygous dominant to heterozygous dominant to homozygous recessive, write out what you're doing so you can find the best answer when it comes to end of course testing. So then phenotypes, we have 1/2 to 1/2 or 50% to 50%. Or you could just say 1-to-1. So now we're going to look at mom and dad here, but we're going to do the same thing here. So what we do is we do the same thing we did with monohypocrosis, okay? Is we're going to go through that law of segregation where those alleles split into gametes. But we're also going to go through that law of independent assortment because these can be assorted separately. So that's where we're going to make it 2 separate problems. So in this case, capital F, lowercase f, capital F, lowercase f, and then lowercase r, capital R, capital R, and then lowercase r. And I think I jumped the gun on what I'm showing you of how we got it. Here's mom and dad. Okay. Here's mom. Here's dad. Dad is heterozygous here. Mom is heterozygous here. Dad is homozygous recessive. Mom is heterozygous dominant. There and there. And so the one we just did was we split them apart. We kept P male here and P female here. So hopefully they just kind of, so what I end up doing is something like this. So I know which one's which, and I kind of cross them like this. So I know I've kept them separate, but they're going soon. In this case, it's my male on the left, female on the right, but also my law of independent assortment. So this is trait 1. This is trait 2. And this is just kind of showing you why we do it the way we do it. Okay. So in this case, there's a 50% chance that capital F is going to be there, 50% chance just like last time. And then this lowercase r, there's a 100% chance. So now we're going to multiply. So I'm going to do this in 2 different colors for us so you can kind of see. I'm going to do let's do green. You can see that. I'm going to multiply this times this. So capital F, and it's 1/2 times 1/2. So makes it 1/4. Then we have this first F again times the second F here. So capital F, lowercase f is 1/4. So now I'm going to change colors again. I think I have to do a blue this time. So now I'm going to do this lowercase f times this capital F. I always write the dominant first so I can see it. So it's 1/2 times 1/2 is 1/4. This lowercase f times this lowercase f is that 1/4. So it's kind of like that FOIL method. You don't, 2 sperm or 2 eggs are not going to fertilize each other. So you're going to multiply it by the opposite gender with the opposite gamete. So then we're going to go through our rules of addition. However, this one and this one are the same, right? So that in this case, we could combine them together to be capital F is 2/4, right? So those are the genotypes. Now the phenotypes is we're going to see anything with a capital F, or anything with a capital F, we're going to add those together. Anything with a lowercase f, we're going to add together. So in this case, anything to do with purple, because purple was dominant, we add this one up and this one up because they both have a capital F in it, make it 3/4. Anything with white, there's only one lowercase f, and so that's 1/4. Well, in this case, we're going to do the same thing. We're going to do lowercase r times capital R. That's 1/2 capital R, lowercase r, and then 1/2 lowercase r. So then phenotypes, we have round, and we have wrinkled. So it's going to be 1/2 round because it's this one has a capital R in it, and then 1/2 wrinkled because it's lowercase r. Okay. So now we're going to do the same thing we did already. I'm doing this from memory. Because I'm going to show you why we do this. Because they're not going to ask you to draw a chart on a multiple choice question. They're going to ask you, what is the probability of having this as an offspring? And I'm going to make this 2/4 just to make it easier for you. Because everything is already in 4, so 1/2 is 2/4. Okay, so what is the probability of an offspring having purple flowers and round seeds? So now what we're going to do is we're going to look at, since it's purple and round, we're looking at traits. So we're going to be looking at phenotypes. So purple is 3/4, and then round seeds is 2/4. So now what we're going to do is we're going to multiply across, right? So it's going to be 16, right? Because 4 times 4 is 16 on the bottom. 3 times 2 is 6, right? And then you can reduce that. And so common denominator is 2. So 6 divided by 2 is 3. And 16 divided by 2 is 8. So there's a 3/8 chance that they're going to be round and purple. And what is the probability of having offspring with lowercase f, lowercase f, lowercase r, lowercase r? So we're looking at genotypes because we're seeing alleles. So lowercase r, lowercase f. We're going to multiply them. 1/4 times 2/4. And we're multiplying because it's the probability of each one occurring. So 2 times 1 is 2, 4 times 4 is 16. And then we can reduce that by 2, which makes it 1/8. Okay? So that's going to be kind of why we do what we do. Okay? And so that kind of gives us that trihybrocross is the exact same thing. We're going to do 3 different traits. So that's the first trait, second trait, third trait. You would go through this probability again, and then I'd ask you, what is the chances of these occurring? You multiply those across, and that's what we do. And so I've also put up a practice document for you that we've already done that 2 weeks ago, but because of spring break, you might have forgot. So I would look at that if you kind of forgot how to do this, just because it's going to be very, very hard to kind of do what we talk about when we talk about pedigrees and the odds of this being dominant or recessive, and what could the parents be without knowing the percentage or the probability of this. Okay. I hope this helps you, and I will see you next time, guys.